Hello and welcome to this video on the basic unit of your muscles, myosin and actin. While this video may not make you swell, it will help your brain get bigger. Myosin and actin are essential to muscle activity. Without them, your muscles do not contract. No contraction means no power. No power gives you a lot of problems. For example, without the ability to contract muscles, you can't breathe. You can't pump blood around your body because your heart does not contract. You can't lift anything. You can't conduct any kind of research. When the myosin head, which is the fundamental unit we're talking about, is in a resting state, it's going to be attached to the actin filament, and by doing so, it effectively acts in a manner of speaking like a lock and key model. This is also why when you die, a rigor mortis sets in. It's the myosin head binding to the actin filament. Without ATP, which life makes by necessity, the actin filament and the myosin head cannot disassociate, and therefore the sarcomere, which is the larger structure of a muscle, will be locked in place. Given that ATP gradually runs out after the human body dies, it's why rigor mortis takes time to set in. Let's assume, for the purposes of this video, that we're talking solely about a living being, as there's a slightly more complicated process involved in the breakdown of the myosin and actin in terms of decomposition and rigor mortis wearing off. For the purposes of this, we assume living animals, as that means ATP is available, and as a result, the actin and myosin can freely interact as required. What you see is that you have the ATP here. The ATP is going to be hydrolyzed, that is, breaking off the phosphorus group, leaving you with ADP, or adenosine diphosphate. The myosin head, because of this free phosphate, can attach or reattach to the F-actin. This F-actin is just slightly further down the filament than where the myosin head originally started. This allows it to contract, effectively pulling it or ratcheting it just a little bit further along. Once the myosin head has moved itself down the actin filament, which is effectively the contraction process, the phosphate will be removed and you now have the first step of contraction. Because the myosin neck, which is what attaches itself to the head of the myosin, has to swivel around due to the translocation or relocation of the myosin, which is what moves it, it will, by necessity, need that phosphate. The reason it needs it is that the phosphate changes the affinity of the myosin head, and because of the change in the myosin head's affinity, effectively the charge on it, that is why it moves. The ratcheting process is made possible. In order to regulate when this is happening, you have a tropomyosin. Tropomyosin is another protein within the muscle. It's a somewhat fibrous protein, and it sits alongside the actin microfilaments. Now, its role is to ensure that until the ATP is broken down into ADP, the myosin binding sites are not accessible, or at least don't have access to the actin. Because of the way it interferes with the, interaction of the two, you don't have contractions until you're trying to allow contraction to happen. This is part of a larger complex called the troponin complex. The tropomyosin and troponin won't be allowed to move and make room for the myosin actin to work the way they need to do until calcium is released. And when that happens, you find that the troponin, which is the structure getting in the way, changes its conformation, and this reveals the myosin. The myosin now, with ATP turning into ADP, can then bind to the actin filament. Because you are only releasing calcium as required and in very small amounts, it allows it to be recycled quite readily. And it arguably needs to be recycled quite readily, because there's not a lot in the end of nerve endings and in the muscle itself. The calcium is located within the endoplasmic reticulum, which is another reason why you need to constantly cycle through it. Part of the process of releasing this is to have within the muscle the endoplasmic reticulum, which is where you have most of your protein synthesized, and within that the sarcoplasmic reticulum, which is a specialization within it, respond to the calcium released from the nerves. Now, it can either be calcium released from the nerves, or in most cases it's going to be something like noradrenaline. Either way, it results in the depolarization of the muscle cell membrane. That muscle cell membrane depolarization causes more calcium to become available. Part of that more calcium is a pump. The pump literally shoves out as much of the calcium as it can within its ability to do so. Along the release of the calcium itself will travel along something called transverse tubules, or T-tubules. These are effectively highways running out of the sarcoplasmic reticulum. They allow for the calcium to be very quickly shunted out and into the area around the myosin and actin. Generally speaking, there are roadblocks along this highway or freeway that would prevent calcium from being able to get out under normal circumstances. But when the nervous system innervates the muscle, these roadblocks are removed, and this is why it can more easily, and in the most cases, freely get out. Once out, it now binds to the troponin C, which is part of the tropomyosin arrangement, which allows for the myosin and actin to now be more freely accessible to each other, and then the myosin head can bind to the actin filament and begin ratcheting. From this, you then have your muscle contraction. You can see how there are multiple steps regulating how and in what way a muscle can contract. Further to the, let's say, active or moving parts of the muscle to contract, there are what are described as static or structural elements to it. These are things like proteins in the sarcomere. One of these is called titin. This is a gigantic protein, and it's a way of fixing the myosin fiber in place, and to an extent, it ensures that although the myosin can contract, that is by pulling on the actin filament, it will always go back to where it started from at the end of the contraction cycle. This ensures that your muscles will always reset, allowing you to have another contraction happen, and to a certain extent be far more efficient. Imagine if you had to consciously both contract your muscles and relax them. It would be very uncomfortable in very short order, as parts of your body would unconsciously contract or become rigid and stiff, and you wouldn't know that they were a problem until it began to become sore and painful. Just in terms of the titin itself, the titin attaches or wraps around the myosin, and it will do so in multiple locations, ensuring that it can always pull back on it. But the locations where you see it binding to it are towards the middle. They're the medial area. Now, this is going to be found near the H zone. Within the H zone, you'll find the Z line. The Z line is effectively where titin is attaching to the telethonin complex. The telethonin complex is the larger structure area here. Because of what it is and what it does, it also interacts with several other structures, and these are things like the obscurin in the I band, myofibrils in the sarcoplasmic reticulum, and in the M band. For the purposes of this video, we won't go into a great deal of detail what they are. Just understand these structures are located here, and they do have a role in this. So far, everything we've described assumes that for the purposes of a contraction, you have a nerve somewhat independently of the muscle itself acting. And this can be true, although rarely the case for most purposes. Instead, what you find is that you have the muscle, whether that is something like skeletal muscle, smooth muscle, or cardiac muscle, and the nerves make a direct connection effectively inside these muscles. From there, they will release their neurotransmitter. This will be something like acetylcholine, for instance. This acetylcholine will bind to the receptor on the muscle cell itself. Now, we mentioned earlier how this results in the release of the calcium. The way this happens is that there are ion channels in the muscle cell membrane. These membrane-located channels can be opened or closed in various ways. One of the ways this can happen is by running a voltage across them. The voltage change causes these channels to open up, and this is what makes the membrane of the muscle more permeable, particularly to calcium. Partly the membrane-bound ion channels, and partly the channels responsible for the cytoplasmic reticulum being able to release the calcium. The more calcium it can release, by virtue of a bigger voltage change or more, let's say, a frequent innervation by the nerve and the resulting repeated stimulation of that voltage change, the more channels that can be opened, the more calcium released, the stronger the contraction. This then starts the process that we described from the very start of this video. This is how you can do the most basic things, like breathe, swallow, or post weird conspiracy theories online and accuse people of random racism. Admittedly, the latter two are not as important as breathing, swallowing, drinking, and the heartbeat, but nonetheless, it's how you can get away with these things and how your body's able to do them. Thank you for watching this video. If you found it interesting, consider liking, sharing, and subscribing. Please do post any comments, questions, or suggestions you have below.